all the girls around the world, love and be loved. From Linda G and Lady T, baby.
I wanna talk about her, Linda good girl, yeah. 아직은 조금만 더 Linda 지치지만, yeah. I wanna talk about her, Linda good girl, yeah. 아직은 조금만 더
Okay, now here now we are about to take a look at a different type of actually the exactly opposite type of the reaction that we saw in the previous session of the lecture. This one is taking place in the chloroplast and the name the photosynthesis. So here what happens is using the carbon dioxide as the initial material to build the glucose, which is now we hopefully easily understand that it's going to be an anabolic reaction from a simple inorganic carbon containing molecule into the organic complex, relatively complex glucose molecule containing 6 carbons and other elements including hydrogen and oxygen. So as an anabolic reaction, the energy source here is being provided from sunlight and any other light source, and the packet of the unit of this energy from sun or any other light is called a photon. Those energy interpreted as the unit of photon is captured by very specific specialized molecule present in on the leaves of in particular actually in the chloroplast membrane of the leaf of the plant, which such name is chlorophyll. So the chlorophyll which are abundantly present on the membrane of the chloroplast. Other one actually capture that are capable of capture the light energy, and then fund those energy captured by them will be funded into anabolic process of glucose synthesis. The structure of the chloroplast is somewhat similar to, which is reminiscent of mitochondria, in that it also consists of a double membrane, so outer membrane and inner membrane. And in the case of mitochondria, inner membrane is highly folded structure as opposed to, in contrast to, the outer membrane. This is mitochondria. But in the case of chloroplast, that inner membrane folding has been far more extensive, in that now, as you can see, this internal, the folding of such inner membrane has been achieved to the level of highly stacked internal sac-like structure, which are actually originated from the extension of the inner membrane. Now we have some different terminology in the case of chloroplast. In mitochondria, we call that the internal space as a matrix. However, in the case of a chloroplast, which the equivalent of the matrix, now internal space, is called the thylakoid. Sorry. This internal space is a stroma. And the inner membrane, as a special name, inner membrane of the chloroplast is called a thylakoid. And in particular, those extensive, very extensive folding to the form of some of a stacked over sac-like structure is a lot of times it's called the grana thylakoid. Once again, all these names are to simply to provide some distinction, and when they were referred, so that you don't get lost what they were being referred. That's the only purpose of mentioning each different parts of the chloroplast. It's not at all about that suggesting you to memorize anything about this different part of the chloroplast, as well as the mitochondria. So those light energy capturing those molecular chlorophylls are highly abundant in the inner membrane of this, the inner membrane in particular, the endogranar, which is an extension of the inner membrane of the chloroplast. So here, light energy, and another very strikingly similar feature of this, the reaction happening in the photosynthesis, is this: the hydrogen ion proton accumulation in the space between the outer and inner membrane, and that potential energy is utilized into the synthesis of the ATP. We have just exactly the same thing seen in the case of the electron transfer channel of the aerobic respiration, which is occurring in the inner membrane of the mitochondria. So the overall procedure is what's in the using, once this ATP is being synthesized, the reason why they need to synthesize the ATP from using the energy coming from a light energy is so that they can use this ATP energy in the anabolic reaction, actual anabolic reaction of conversion of the carbon dioxide into the glucose. So here, the cartoon summarizing this entire procedure is a nicely drawn figure. The only reason why I mentioned this nicely drawn figure is because I myself generated this figure using Adobe Illustrator. So here, as you can see, the buildup of all these hydrogen in this small space. By the way, actually, this small space between outer and inner membrane, and both in mitochondria and chloroplast happen, is a very good example of the compartmentalization, so that using very small particular area generated by internal membrane, the organelles, and the small spaces generated by such different organelles are functioning very specific ways. And as we can easily understand, okay, so those the mitochondrial space between inner and outer membrane space, which is being utilized in the accumulation of a hydrogen ion, so that with which the ATP is being synthesized, and the same thing is true for the space of the chloroplast, is such an example. And another thing is there are photosynthetic bacteria. Remember, this photosynthesis, and as a result release of the oxygen, so that resulting in the accumulation of oxygen in the atmosphere was the triggering point of all this evolution of the eukaryote and the multicellular organism in the early evolutionary stage of the Earth. So then those bacteria, prokaryote, do not have any organelles. And then how do they manage the photosynthesis? Well, they use their own some space separation from the cell wall, between the cell wall and the cell membrane. Using that, they kind of similar to the space between outer and inner membrane of the chloroplast. They perform their respective function of this the photosynthesis. And the same thing is true. A lot of the bacteria also perform the aerobic respiration. Even they don't have any mitochondria, but still they do have this aerobic respiration, having all the same enzyme complex present on their membrane, cell membrane, because they can utilize their space between cell wall and the cell membrane and then do the exactly virtually identical function as we have seen in the case of this electron transport chain. So anyway, so this one is so in this overview, what we can understand here, the key point is, in photosynthesis, the light energy at first being converted into the ATP synthesis. Some might wonder that why, like also plant cell have the mitochondria, so then why don't they use the ATP generated mitochondria to support this reaction? Why do they have their own system of ATP synthesis? This is a very good question, but probably one reason is because actually their origin of the metabolism is too separate, too independently. So this one actually also reflects the fact that the endosymbiosis origin of the prokaryotic, the origin of this chloroplast and mitochondria are also being supported by this fact. And another thing is ATP is very difficult chemical to easily move them around across the cell membrane. So it's much better to have their own synthesis of ATP where on site where they are needed is another probably the explanation, although that is not actually entire 100% reason why this ATP synthesis has to occur in the chloroplast. But anyway, that's probably as best answer or explanation I can think of if you asked. So anyway, this ATP synthesis driven from the sunlight is the first stage, and the second stage should be using this ATP. Now you are actually, this is a real purpose. The second stage is your purpose. Now you synthesize the glucose from carbon dioxide. It's two separate procedure is what basically what it is about the entire process of photosynthesis. So if you zoom in first stage of this photosynthesis, that has a specific name of light reaction, because that is the only reaction of the photosynthesis absolutely requiring the light energy. So what happens in this light reaction is, as I have already explained, it is extremely, in essence, very similar reaction to that happened in during the electron transport chain during the aerobic respiration in the aspect of accumulation of hydrogen ion build up within this small space now drives the synthesis of the ATP. And another thing is also the ATP synthase, the same exact ATP synthesis enzyme also is present in the inner membrane of the inner membrane, which is a thylakoid, inner membrane of the chloroplast, and that is the tunnel through which hydrogen moves, and then during which ATP is generated. So this another thing is the very similar electron carriers, the cofactors that we have seen, like NADH and FADH2, similar to such electron carriers, also are present in this light reaction. And what's happening is it is actually this oxidation reaction of all these electron carriers. And who is the one actually the one finally reduced by this electron transfer reaction, which is an oxidation reaction and oxidation reduction reaction? In this case, well, we don't really have to go that far because it's too much for for this level of lecture. However, instead of an oxygen, in the case of the electron transport of the aerobic respiration, the final electron receiver who is finally reduced was oxygen. But in this case, the one of the electron carrier, the one NAD, the similar to NADH, is the one finally receiving all this electron to become reduced form, whose name is NADPH. Okay. So that one is the only thing that I am mentioning this is here, although the same electron transport chain, identical, kind of virtually identical electron transport chain reactions is taking place, but the final electron acceptor is different from the case of the electron transport chain in the aerobic respiration. That's the only point. And also another thing is, then who is the one first being oxidized? Here the interesting thing is water is the molecule that initially being oxidized. That means the electron present in the water is stripped up, being taken to initiate this. So the beauty of this special thing, not beauty, maybe the special thing about this water oxidation is, chemically speaking, it is extremely difficult reaction to carry out. So for that, this energy is initially required, or also the sunlight energy and another light source is required. So once you break this water molecule into two to get their electrons out, then the end result is now you have an oxygen and hydrogen ions are being separated. So that is the reason why you have this oxygen released, and as a result we have a plane of oxygen around in the air because of this reason. So actually this particular same type of a chemical reaction, human only very recently, relatively recently, were able to learn and imitate this reaction. As in these days, before in the submarine, they had to carry the whole huge oxygen tank so that provide this breathing air to the crew members. However, in these days, a lot of those, usually most of this submarine, they can generate their own oxygen by exactly the same chemical reaction of splitting this water, so that as a result, oxygen can be released. That's another thing that we have learned from this photosynthesis. And the place of this reaction, the inner membrane, is very reminiscent of that aerobic respiration final stage, isn't it? So inner membrane of a chloroplast. The reason why it's taking place in the inner membrane is same thing. Those enzymes that are performing these electron transfers are present, embedded in the inner membrane of this chloroplast. So here, bottom line is water is, as I said, water is oxidized, and as a result, oxygen is generated. So this is a simple overview of such a chemical reaction happening in here. So now, once as a result of the light reaction, which will end up in generating this ATP through the identical mechanism of what we have seen in the aerobic respiration, and also this NADPH, which is similar or actually identical to the NADH that appeared in the aerobic respiration. These two will be used as an energy source in the second stage of this photosynthesis, which is also surprisingly, this reaction, the second part of the photosynthesis reaction, is organized in the cyclic form of the reaction. The only different thing is its name is a Calvin cycle, and as a result, this precursor of the 6-carbon glucose will be generated. So that reaction is called dark reaction because now this reaction itself does not require the presence of any light. Once you have generated this ATP and NADPH from the light reaction, the light does not light is not required. So as I said, its name is called the Calvin cycle in honor of the biochemist who firstly discovered and figured out the entire chemical reaction. This is the one. So now you generate this precursor form of the glucose, and eventually glucose is produced within the internal space. By the way, what did I say about the name of the internal space of the chloroplast? That's stroma. And now that glucose is being assembled into the polymeric form of this glucose, starch. So starch is actually individual. So in the starch molecule, this ring structure is indicate each individual glucose molecule, and several hundred and more glucose molecule assembled into form of one starch molecule, and which is a very ideal form of a storage. And actually in our animal cell, in particular in like liver, a very similar storage form of the sugar, glycogen, is actually almost identical in chemical structure to the starch. So both starch and glycogens are the storage form of this sugar, simple sugar glucose. And also, once you have done this, now some of them can be funneled into some other different types of metabolism to synthesize another different type of sugar, sucrose. So this sucrose is a transport form of this sugar. If the starch was the storage form of the sugar, then sucrose can be said of the transport form of the sucrose. In other words, as a single plant, only aerial part, in particular those leaf area, also the healthy young leaves, not too young, these areas are the only area where the photosynthesis can occur. So in other words, only those areas having the plenty of this photosynthetic apparatus in the form of chloroplast can perform and synthesize the sugar. What about the non-green part, in particular this root? They're not able to carry out their own photosynthesis, so they can be easily very hungry. So in support of those areas where they cannot their own photosynthesis, their photosynthetic product has to be transported into those areas where they can't. So in what kind of form of the sugar are they going to transport this? Glucose, if they simply ship their product as they are, the glucose, and it's too dangerous, too risky, because glucose is, chemically speaking, is too reactive. So it will easily interact with other chemicals to become something else, or easily metabolized. It's all gone. To prevent that from that happening, so plants convert their product into sucrose, which is actually 2-carbon sugar, as you can see over here. So this sucrose is the ideal form of the transport sugar, and then they are being shipped into those areas, and later once they arrive, they are readily converted back into the glucose, and then depending on who you are, like if you are like potato, then you also convert them into their own storage form of starch. So that's what we have take advantage of in the form of potatoes and all kind of related products. So all this dark reaction in a nutshell can be said of this carbon fixation reaction. What do you mean by this carbon fixation? This atmospheric carbon dioxide is being fixed into the plant system to become more complex form of the sugar, like glucose. So to that matter, energy is required. So the form of this ATP made from light reaction is used. So here, these two figure compares the architecture, organized architecture of chloroplast and mitochondria, and utilizing their similar architecture in the very similar tasks, like building up of this hydrogen ion to drive this ATP synthesis, and same thing also the small space here, hydrogen ions are accumulated, and then now ATP synthesis can occur there. So that's the whole essence of respiration, aerobic respiration, which requires the oxygen and the photosynthesis. Once again, these are each of them very good representative of typical like anabolism and the catabolism, respectively, and so we can say they are actually in direct opposite reactions. Like if we try to sum up, in the case of the catabolic reaction, which is aerobic respiration, starting from glucose, and to do so, to carry out this reaction, absolutely you require this oxygen and become this carbon dioxide and water, because oxygen will be reduced into water, and as a result energy is released, and that energy is used in synthesizing ATP. Okay, now in the case of photosynthesis, which is totally opposite direction, so starting from this carbon dioxide and water, in the water is used in the light reaction, remember, to split this water molecule to generate. So because of that, oxygen is produced, and carbon dioxide is needed in the second stage, which is dark reaction, and this carbon dioxide is fixed into glucose molecule, and so that's the summary of photosynthesis. Now, the final section of this chapter 3 is about internal skeleton of the cells. Each cell, each eukaryotic cell have the internal skeleton system, just like our body is being supported by structurally the bone, the skeletal system. So they are collectively called the cytoskeleton. And these are the cytoskeleton system is maintained by 3 distinct big proteins in the eukaryotic cell, which are they are one a microtubule and another one is a microfilament. And usually this microfilament is called actin microfilament. That's and microtubule and intermediate form of intermediate filament. All these 3 proteins are long rope-like, tube-like proteins. So, like tubules are some hollow. The tube-like structure is usually called a tubule. Whereas the filaments is called the thread-like structure is usually called a filament. They have such names because their appearance look like either tubule or filaments. And microtubule is the largest, the biggest, and the actin microfilament is the smallest. And in terms of the size, the intermediate filament is like in the intermediate size. And all these 3 different types of protein, long proteins, comprise of all network-like system inside of the cell, so that provide many different functions. Another interesting thing about this cytoskeleton is until very recently we have thought that this is the cytoskeletons are the specific feature of eukaryotes only. So in other words, the prokaryotes, like bacteria, don't have anything like this cytoskeleton system. Wrong. More recently, it's been discovered that actually the bacteria also have their own kind of version of their internal structural supporting system, very much like the cytoskeleton system of the eukaryotes. So we cannot say anymore that the cytoskeleton is the feature that only exclusively can be found in the eukaryote system. So all this knowledge obtained, the information obtained in the life science is keep changing every minute, actually, as a result of the new discovery and the corrected opinion and then corrected information. So maybe that's one of the beauty of this life science. Maybe not only the life science, but any other all this scientific area is the actual common characteristic of such a dynamic nature, or keep changing. So anyway, the important thing about this is rather than trying to minding each specific details, like actin consists of several actin subunits or whatever, blah blah. These are the main thing is all these 3 different types of cytoskeleton, the proteins that are used in building the cytoskeletons they are all long tube-like or thread-like proteins. Okay, then to understand how do they make such tube-like or long thread-like protein structure, then one can think of how do they build? Probably it's the only way in a living system. They don't have any kind of big template to print any big structure, unlike a human-made factory. So the only way they can assemble such a big structure is starting from one small thing, the subunit. So the same principle of a building polymer from starting from small simple monomer that also applies to this example. So each different small units come together, being assembled to make such a big structure is always the same case, whether it is in the case of actin microfilament, or in the case of intermediate filament, or in the case of microtubules, or anything, the same thing. And also, all these colored, these are all the fluorescent micrograph photographs, and all these different colors are generated by specific labeling of a fluorescence, specifically picking on. Here, in this case of the actin proteins, are all in red colors. This is the general shape of overall typical animal cell, which is actually a fibroblast, connective tissue cell. And here the green things are all picked up by this green fluorescence, so to indicate how extensively these are spread within a single cell of an animal, and this intermediate filament. And same thing goes for this microtubule. So in other words, all these three different types of cytoskeleton proteins are spread literally like a spider's web-like complex and interlocking networking systems are the one that the cytoskeletons are being the way they are organized. So what's their function? Many functions are actually provided by this cytoskeleton system. One is maintaining the overall shape of the cell. We may actually take a certain shape of an animal or plant cell for granted, but especially in the case of animal cell where we don't have the rigid boundary provided by cell wall. We don't have any animal cell, don't have any cell wall. So actual unique, distinct shape of different types of animal cells are the result of the specific pattern layered by specific cytoskeleton protein. For example, like typical appearance of nerve cell, neuron cells are like this. Here, nucleus is there. How did this the neuron cell, the nerve cell, have this type of a very unique, interesting looking shape? These are all due to some specific arrangement of individual different cytoskeleton proteins is the one. On the other hand, another type of animal cell called the epithelial cell looks like this usually. I will always try to jokingly compare this epithelial cell as a Bart Simpson-like cell. At the bottom is flat. The other area, it's like a finger-like projection. These are all the result of different arrangement pattern of a cytoskeleton system. So maintain the shape. Another function, important function is organelle. Within each different cell, different organelles, instead of freely floating around anywhere, usually particular organelle have its own specific fixed position. Just like within the nucleus, chromosomes and chromatins, each different chromosome have their own unique territory, as we already went over in previous lectures. Similar thing is observed in the case of organelles in the cytoplasm, and the reason why they can maintain such relative specific position is due to their fixing things. Position is fixed by the attachment from the specific cytoskeleton proteins. And also a more important thing probably is it provides the road tracks, railroad tracks, or maybe you can compare it as a highway for all those movement of transport vesicle. Where did you see this transport vesicle? They are the one who actually carrying out those cargo movement in the case of exocytosis, and also similar vesicles are involved in the case of opposite way of movement from external to inside, the endocytosis. So all these the small vesicles, you may have thought that those vesicles are just like balloons, so they virtually float from one place to the, for example, like if one transfer vesicle is to move from endoplasmic reticulum to Golgi, they will simply float around just like they are on the water. No, it's not. Actually, they are walking. Each transport vesicle is walking through on those specific cytoskeleton protein, as if that protein is a kind of a road upon which this transport vesicle can walk. Is extremely important function, isn't it? So such different functions are summarized in this diagram, which is describing such particular epithelial cell, as I mentioned. The one and this finger-like projection is actually maintained by here this red thread-like things indicated actin filament here, and this blue, sky blue color of this represent microtubule, and this dark blue rope-like structure is the intermediate filaments. And so roughly and simplistically they are usually arranged like this, and as you can see along with this each different microtubule is being utilized as if like they are highway or railroad tracks on which different transport vesicles and even sometimes the organelle itself, here it looks like a mitochondrion, moving on each. So in other words, all these different transport vesicle, or in some cases the organelles, are not moving from one place to the other freely, but they have to exactly follow on the path as a track. And so these are the kind of, also as you can see, the reason why this Golgi is usually placed around on this position is because this one main pivot of this microtubule is trying to push this Golgi area to a specific location so that they cannot easily move around to other places. So that is also a very important function, as you can easily see. And here it's describing one end of this nerve cells, a neuron. So usually here the nerve signal, as we will see in the later this semester, transmission of nerve signal will travel from this, like following this arrow direction, and so this long extended structure of the nerve cell, which is maintained by its shape, is maintained by the combination of these different types of long microtubules as well as intermediate filament, along which these structures. So here, the nerve signal, the reason the nerve signal can travel like this is because some specific chemical, as we recognize as the neurotransmitter, the chemical, as we will be seeing later on, are actually traveling along with this track, and then will move from one nerve cell to the other. And another very important function of this cytoskeleton is in the control of extracell division. So speaking of the cell division control first, so as we can see over here, once again, this sky blue like color is representing microtubules. So this is like a cell is about to divide into two. So one cell, the way usually or typical animal cell divides is now at the imaginary center, the equator, cell will become like this, and then eventually split into two. This is how usually the animal cell is being divided. And to do so, there are two events must be carefully taken care of. First, during this cell division, your genetic material chromosome should be segregated into equally. So the same half the amount of the entire your genome must be split into one end chromosome, and another set of chromosome has to go into the other one, so that they can end up both the same amount and the same kind of a chromosome in each dividing the new cell, the new daughter cell. How do you accomplish those exact separation of each chromosome? Here, each red thing represent the chromosome. To each chromosome, this microtubule attached, just like a lasso tool. So you pick up using this microtubule rope to each chromosome, and then hook, and then pull. So that's how you divide each different chromosome into two, the opposite end. And for that matter, this microtubule, this particular type of microtubule used in the separation of chromosome is called, its name is spindle fiber. So this spindle fiber is made of microtubule, is the thing that you might want to carry in your mind. And next thing is that's one thing, but also your whole entire cell, which like your whole entire cytoplasm also has to be divided into two halves. How do you succeed in such a task, which is not easy? That one is accomplished by squeezing this center. Imaginary center is, as you can see in this diagram, is being squeezed and pinched like that. Yeah, that is the term we can describe it. It's pinched in, so that if you keep squeezing at a certain area, the central area, then now eventually it will become like they are disconnected to become two separate independent entities. That's how you do. And how do you manage it? Such task by squeezing and contracting using a specific type of a protein, which in this case actin. The actin microfilaments are arranged at that point and then being contracted. And one thing interesting is also when we actually do our muscle flexing, which is actually identical, is nothing but contracting our muscle fiber. And those muscle fibers are made of the same, this actin microfilament. So in other words, what we can take from these two is actin microfilaments are actually in function. They are specialized in contraction. Anything contraction is needed, I will be there. So similarly, when you try to contract to squeeze in the cell, animal cell to part ways into two entity, yeah, I will be doing that job. And also when you try to move your body by this muscle contraction, also I will be needed then too. So that's two functions that are respectively provided from microtubule and actin filament in this dividing cell. And also cell movement, cell motility is so like in the example, for example, in the case of amoeba, each different surface area will constantly move up and down or back and forth, however you might want to describe, so that this cell surface usually some kind of projection. So this extended projection and contracting, withdrawing projection structure we call as a pseudopodium, like a false legs. That's how you extend your surface and then capture some of the prey and then internalize this. And those are all provided by rapid synthesis of internal actin microfilament and then rapid degradation of that once built microfilament. So that's how you accomplish extension and withdrawal of a particular surface. The hair-like structure is also made of microtubule, and this hair-like structure is utilized in the movement of, and similar hair-like structure is also present in the respiratory tract, so that they can help generating some the liquid flow through this respiratory tract and the other digestive tract too. And the single cell also can move using the projection-like structure, which is also made of microtubule too. So overall, instead of like focusing on little details, whether it's a microtubule or actin microfilament, they also can be utilized in providing movement of a single cell too. That's the bottom line. And once, as I mentioned, this muscle contraction, which is provided by the same actin microfilament, is another example of such important function provided by the cytoskeleton. So perhaps that's probably the kind of digested summary of all those things, main things about the cytoskeleton. So with that, I think this chapter 3 can now be finished off. So then next, although it is a relatively short lecture, I will not go over into any other lecture, new material over here, but the new set of the chapter 4 lecture will be done by in separate lecture series. So for now, see you in next lecture.